Hi, and thank you for clicking on my poster. My name is Mathias, and I'm a PhD student in the lab of Caroline Dean at the Nielson Center in Norwich, UK. In the Dean lab, we're investigating an important question of how plants know when to flower. The Dean lab has been studying this for the last many years, and it comes down to one key gene and the regulation of this gene. And this gene is called FLC, short for flowering locus C. And FLC you can think of as the brake on flowering. So when FLC is highly expressed, as in the autumn and early winter, it prevents the plants from flowering. However, as a response to cold and during winter, FLC is switched off, and this exposure to cold is remembered in the spring, allowing the plants to flower. But how do plants sense and remember winter? This is the big question of the lab over the last 30 years. And this comes down to FLC and how FLC is suppressed by a very conserved protein complex known as PRC2 or Polycomb repressor complex 2. PRC2 is well known for its role in developmental regulation in many organisms, both in plants and also mammalian cells. But over the last couple of years, it has been clear that not only is PRC2 important, but also the proteins that are associated with PRC2. In plants, one of these proteins is called Win3, and Win3 is induced as a response to cold exposure. So as plants experience cold, Win3 is induced and associated with PRC2 and switches FLC off. And this is this complex that I'm interested in my PhD. And first, to show a little bit of background in these proteins, is that when you give the plants cold, this complex of core PRC2 shown in purple and these associated proteins Win3 and Win5, which are homologs and very similar, they come together, as you can see with this protein IP mass spectrometry experiment, where we give the plants no cold or 6 weeks of cold, and you can see that only after 6 weeks of cold this complex comes together. However, how exactly this complex comes together and how it goes to chromatin and FLC, we still don't really know. So this is what I'm particularly working on in my PhD. And we are taking a very interdisciplinary approach by combining molecular plant biology here at the Nielson Center with more structural and biochemistry work at the MRC Laboratory of Molecular Biology down in Cambridge. And our analysis so far has shown that this protein complex is a lot more complicated than we first thought, and we've now only focused on these accessory proteins Win3 and Win5, which we can think of as being more or less identical. And our analysis has shown that these proteins contain several novel domains that we didn't know they contained before, which start looking in them into more detail. And you see a schematic of the proteins here on the right with their domain architecture, with 4 very closely arranged domains which we think are involved in chromatin targeting or protein-protein interaction, which is this NZF, PhD, Winkillix, and FN3 domain you see as one combined superdomain we think of, and then the C-terminal domain, the well domain, which have given names to these proteins, which we call the well proteins. So what we do here during the center, what I'm mainly doing my PhD, is making mutations in these domains, deleting them, or making permutations and putting them back into plants. And we are very lucky that we have a very clear phenotype, and I have tried to show you the schematic that in the Win3 and Win5 mutant, which you can also see here, these are very late flowering and only flower after 2 to 3 months. However, if you give them back the functional well protein, they will be able to flower much quicker after the exposure to winter. This gives us a very excellent assay to assay the functional importance of these domains. So by putting the proteins back, either wild type or with deletions or mutations, we can ask if they rescue the mutant phenotype, if they are non-functional, or if they give us some unexpected results as being later flowering, for example. Now this is a very big project that is currently ongoing. However, we do have some results for the final domain, the well domain, as you can see here by some CoAP analysis, where we show that the well domain is absolutely required for the association of Win3 with Win5. And this combined structural analysis has shown us that the well domain is not only a protein-protein interaction domain, but it's also a polymerization domain. And and this leads us to a model where the well domain acts as molecular Velcro, binding together several PRC2 bin-tree complexes and holding them at the chromatin, allowing them to switch FLC off. Thank you for listening to my presentation of this poster. I hope it gave you a brief introduction to my PhD project, and I hope you enjoyed this poster session. Thank you.